Hey everyone, welcome to today's Dr. Van Arthy's monthly podcast, where we empower comparative analysis of circulating tumor cell, CTC, organoid vs CTC spheres knowledge for transforming cancer treatment. CTC spheres. These are three-dimensional aggregates of circulating tumor cells, CTCs, that form spontaneously in culture under specific conditions. They represent a simplified model of tumor cell self-aggregation. CTC organoids. These are more complex, three-dimensional structures derived from CTCs that mimic the architecture and cellular diversity of the original tumor. They can incorporate extracellular matrix and other cell types. Similarities are derived from patient CTCs, offering personalized insights. Provide a three-dimensional culture environment, more closely resembling the in vivo tumor microenvironment than traditional two-dimensional cell cultures. Can be used for drug screening and biomarker discovery. Enable the study of CTC biology and behavior. Models for studying cancer metastasis and drug resistance. They both, in vitro both CTC spheres and CTC organoids offer valuable advantages and disadvantages. CTC spheres advantages: relatively easy to generate and culture. Cost-effective. Useful for high-throughput screening. Disadvantages: lack the complex architecture and cellular heterogeneity of the original tumor. May not accurately represent the in vivo behavior of CTCs. Limited long-term culture stability. CTC organoids. Advantages: more closely mimic the original tumor's architecture and cellular diversity. Better representation of in vivo CTC behavior. Suitable for long-term culture and complex experiments. Disadvantages: more challenging and time-consuming to generate and culture. More expensive. Lower throughput compared to spheres. Can be difficult to maintain genetic stability. Applications in research. Drug discovery. Both models are used for screening potential anti-cancer drugs and identifying personalized treatment strategies. Biomarker identification. Analyzing spheres and organoids can reveal biomarkers associated with drug resistance, metastasis, and disease progression. Understanding metastasis. These models help researchers study the mechanisms of CTC dissemination, survival, and colonization at distant sites. Personalized medicine. CTC organoids, in particular, can be used to predict individual patient responses to therapy, leading to more tailored treatment approaches. Basic research. Provide platforms for in vitro studies of cancer biology. Both CTC spheres and organoids are valuable tools in cancer research, each with its specific strengths. Here's how they're applied. Key findings and future direction. Research using CTC spheres and organoids has led to several important findings: identification of novel drug targets, discovery of mechanisms of drug resistance, better understanding of the role of the tumor microenvironment in metastasis, development of more effective personalized cancer therapies. Future directions include improving the methods for generating and culturing CTC organoids, developing more complex co-culture models that incorporate immune cells and other stromal components, and using these models to develop new strategies for preventing and treating cancer metastasis. Summary. This document provided a comparison of CTC spheres and CTC organoids, two important in vitro models derived from circulating tumor cells. While CTC spheres are easier and more cost-effective to generate, CTC organoids more closely mimic the complexity of the original tumor. Both models have significant applications in drug discovery, biomarker identification, understanding metastasis, and advancing personalized medicine approaches in cancer treatment. If you are interested in our Australian IP methods, please book and schedule a professional consultation appointment by following the description below. Thank you for watching. That's all for today's podcast. Please join us next time, as I will discuss the details of CTC spheres and organoids culturing information as a powerful tool for new drug discovery. Until then, stay healthy and happy.